Thank you both for your presentation. My name is Dr. Loudig. I am a scientist at the Center for Discovery and Innovation, and I've been working with Dr. Steifelman, Bazralian, and Munver for now a couple of years, and we have set up studies, and I'm really excited about sharing some of the aspect of the study that we are conducting and give you some information. So as you clearly saw, the Department of Urology uses state-of-the-art technologies to treat its patients. At the Center for Discovery and Innovation, and more particularly in my laboratory, we're using novel technologies to see if we can improve early detection and screening of prostate cancer. How do we do that? So one of the important aspects of this presentation is to introduce you to something that we call exosomes. You probably don't know about it, but I'm hoping that by the end of this presentation, you will understand what are exosomes. So here, I'm going to show you how exosomes, which are nanoparticles that are found in the blood, do and what advantage they can provide. So here, you will see exosomes being formed inside this tiny endosome here. Those are the little exosomes. They are formed inside the cells, and there's a fusion of the membrane that produces them for release in the circulation. They contain information, DNA, RNA, protein, and this information is inherited from the cell of origin. Interestingly too, they have surface protein that gives an identity of the cell where they're coming from. Here you see an exosome that has been circulating that enters a recipient cell. Here is an example of a cancer cell producing exosome, releasing those exosomes in the microenvironment or circulation, and changing the fate of a neighboring cell. So what you need to remember when you look at exosomes throughout this presentation are a few facts that I'm going to name here. First of all, exosomes, like I said, are released in the circulation. As I said also before, they have protein on their surface that are inherited from their cell of origin. Exosomes also contain a specific set of information from the cell of origin, an encapsulated portion of information that is retained in the exosome. When they circulate, it's not released, it's not changed, so it makes it a great target because you can remotely, if you access this information, have some idea of what was produced in the cell at the time it was released. Exosomes are produced by most cells, normal cells, cancer cells, and most of the cells of the body. Exosomes are secreted in a larger number by cancer cells, which is an advantage and an inconvenience for the disease, but it's an advantage because they are more detectable than normal exosomes. Exosomes, as I showed you on the last few images of the video, enter recipient cells. They enter the recipient cells with specific marker on the surface that allows them to be addressed to a specific cell, so we can purify them. Exosomes from cancer cell contain different information. That's what makes them more interesting, because we can, when we look for cancer cell, determine the information that we are after. Exosome can be purified using antibodies to target their specific surface protein. So how do we purify and identify exosome from a pool of circulating exosome? Well, simply because they have specific protein that inherited from their cell of origin. And if we know this protein, we can use antibodies that are molecules that capture specific protein, and we can purify them. So now let's look at prostate exosomes, and it's called prostazones. Prostazones are synthesized in the prostate, and they are released in the seminal fluid, and solely in the seminal fluid in healthy individual. When you look at cancer, now you have a tumor with a vasculature that is leaky. So some of these prostazomes that are synthesized in the prostate have the ability to enter the circulation. So now in a cancer patient, you have prostasome in the circulation that you shouldn't have. So with a proper tool, if you are equipped to purify those exosomes, you can potentially diagnose prostate cancer without any other test. So here is a more intricate picture of how we actually run this assay. If you look at the top of these slides in blue, you see what I described so far as in prostazomes. This prostasome contains specific protein, like I said, and you have on the left a list of 4 of the major proteins that identify prostasome from other exosomes circulating in the blood. So we use antibodies to recognize these specific surface markers. And once we purify those prostasomes, we can extract the information that it contains because this information comes from the cell where they were secreted from. So we can potentially look at a cell's content without having to access it. So on the right, you see what we call a heat map. And this heat map contains a list of genes. Each little square is a gene. The color of the heat map determines the intensity of expression. So within one experiment, we can see the expression of thousands of genes and determine, based on the profile of expression, if we're dealing with a normal cell or a disease cell. So just for the purpose of this presentation, I put a tumor prostasome at the bottom in purple. And as you see, there's a question mark because we are still trying to figure out if by looking at prostasomes circulating in the blood, we can capture and identify specific information. And that's what we're trying to achieve in my laboratory. So how does the assay work? The assay is pretty simple. You have a magnetic bead with an antibody that captures all exosomes circulating in the blood. That's the first step. Second, you use that combination of antibodies. Each of these antibodies have a little piece of DNA. And when they are in the proper combination, they hybridize to a piece of DNA that contains fluorescence. If you have 3 signals of fluorescence here, they are yellow, red, and green, then you know you have identified a prostasome. And we have something called a sorter that allows us to separate prostasome from other exosomes. So then we can extract information and look at their content. And that's what we're doing in my laboratory. This assay was designed by a collaborator at Uppsala University. So what is the study that we have established with the Department of Urology and Dr. Steifelman, Baziralian, and Munver? This study includes 600 patients, or 600 individuals, 300 that are controlled, that are healthy, and 300 that are patients that have been diagnosed with prostate cancer. Because the purpose of this initial study is to determine if this new assay is reliable and we can pinpoint the samples that have signs of prostate cancer. So we will collect blood, we'll separate the plasma, we will perform the assay, and we will see if we can accurately diagnose prostate cancer. Once we have the prostasome purified, we will extract the information and perform next generation sequencing to identify if there are specific biomarkers that are associated with different stages, different type of tumors. The idea is to stratify patients without having to do painful biopsies. So what are the goals that we are aiming to reach in the next year? We're trying to complete this pilot study, apply for federal funding at the National Cancer Institute, so we can validate the molecular assay in the Department of Urology at Hackensack University Medical Center. So we can establish a state-of-the-art prostate cancer detection assay in the Department of Urology. I wish to thank all the people that have helped so far, and I'm hoping that this study will soon reach completion.